2. Neurodegeneration. Due to long-term use of aspartame, studies suggest that aspartame and its metabolites increase the risk of neurodegenerative diseases, such as Alzheimer's disease, Parkinsonism, multiple sclerosis, and brain tumors. Methanol, aspartame metabolite, causes increased levels of free radicals resulting in damage to the cell membrane, caused by peroxidation of fatty acid in the phospholipids, damage to cellular components such as nucleic acid lesions, as well as gene damage and repair resulting in apoptosis or necrosis. Moreover, aspartame activates various calcium channels in neurons resulting in cell death. In addition, elevated free radical levels decrease enzyme activity in the liver. Aspartame intake also results in elevated H2O2 levels, placing added oxidative stress on cells. This has been confirmed in studies recording elevated nitric oxide and lipid peroxidation levels. After a 90-day diet with aspartame, mitochondrial oxidative stress leads to apoptosis of adrenal and brain cells. Long-term administration of aspartame has been found to result in degenerative changes in the sciatic nerves, including demyelination, disruption and splitting of myelin lamellae, lamellar structure deformation, and myelin loop formation, as well as irregular thickening of myelin sheaths. In addition, other less frequent axonal changes can be observed. Axons can be shrunk, compressed, and distorted. Their mitochondria can be swollen, as well as RER dilatation and vacuolation of Schwann cell cytoplasm. Aspartame also appears to have a negative influence on the cerebral and cerebellar cortex. Long-term use results in altered glutathione peroxidase and glutathione reductase activities and has been found to affect the hippocampus. Its metabolites, aspartate and methanol, also influence the signaling pathway associated with memory, resulting in neurodegeneration and memory loss. Aspartate acts as a glutamate agonist at the NMDA receptor. Calcium influx activates calcium calmodulin-dependent protein kinase 2, CAMKI. CAMKI induces CAMP response element binding protein, KIRI-BP pathway. Methanol inhibits phosphorylation, while aspartate is an excitotoxic substance which interrupts homeostasis and modifies neuronal cell function. Persistent aspartame intake also results in a decrease in hippocampal acetylcholinesterase activity. All these mentioned factors affect learning skills and memory. Aspartame is also amyloidogenic, being able to spontaneously assemble into an amyloid-like nanostructure, with more intensive aggregation observed at higher aspartame concentrations. The aggregation is characterized by the formation V-like structures. Moreover, existing aspartame fibrils can induce formation of amyloid-like structures from other molecules such as proteins and peptides. The structure formed by aspartame can initiate V-sheet aggregation in the AV140 peptide. This process leads to the formation of AV-amyloid fibrils, which are associated with Alzheimer's disease. Phenylalanine, PY, is needed to synthesize tyrosine, dopamine, noradrenaline, and adrenaline. However, Phi demonstrates strong affinity to large neutral amino acid, LNAA, carrier systems, thus competitively inhibiting other amino acids which rely on this carrier system, tyrosine, tryptophan, and preventing them from crossing the BBB. After aspartame intake, plasma phenylalanine level is increased, thus influencing neurotransmitter concentration in the brain. High levels of plasma Phi inhibit tyrosine and tryptophan hydroxylase, the latter of which is necessary for catecholamine synthesis. A decreased catecholamine level can lead to many disorders. Aspartame can be identified as chemical stressor, since after aspartame intake corticosterone and adrenocorticotropic hormone levels in plasma are elevated. Elevated adrenal corticosteroid level leads to decreased 5-hydroxytryptamine function and could contribute to a depressive state. Moreover, elevation of corticosterone level significantly reduces the weight and size of spleen and thymus.